Thank you. All right, how you all doing? Pretty good? Good. So it's an honor and a privilege to be here, and thank you very much for the invitation. I'm really excited to tell you about our work in de novo protein design. And I can remember quite clearly the first time I saw a protein. It was in a biochemistry class, and it was lysozyme, and I thought, man, someday I want to design an enzyme. And we're getting there. I think, as you'll agree, that we have some very interesting enzyme-like proteins that I'll tell you about today. A couple weeks ago, David Baker had a little meeting in which we talked about various aspects of de novo protein design, and he asked me to talk about the foundational years before Top 7 and Rosetta. And so I'll share a little of that with you, but what I'm really excited about is telling you about what we're doing now. Okay, so Anfinson's hypothesis, we've heard, means that basically all the information that is required for the structure and function of a protein is encoded in its amino acid sequence. And when we started to think about de novo design, we realized that we had to move this in reverse. We start with a function, we build a structure around it, and then we say, hey, what is an acceptable sequence that will stabilize the structure and sometimes its dynamics? As we heard from Alan, it was believed this was an impossible problem. People would say there's 20 to the 100th sequences, the conformations were 10 to the 100th, the temporal problem was impossible. So they raised arguments that this is more than the atoms in the universe, and this just didn't seem right to me. We have 26 letters in the English alphabet, right? But that doesn't mean that we have to enumerate every possible combination of characters to derive the meaning of a sentence. So proteins did evolve, life happened, so it didn't seem that it could be that hard. So we took a minimalist approach to this. As you know, simplicity is the ultimate sophistication, to quote Da Vinci. And we also, though, have to keep in mind from Einstein that we want to make things as simple as they possibly can be, but not simpler. So our approach, I think, has illuminated principles underlying the structure and function of proteins, and as you'll see, has led to therapeutics and catalysts. So when I started this work, I had just started at the DuPont company, and my job was to make peptides for whoever asked for a peptide. It was kind of like when I had worked in a restaurant kitchen, and my job was to chop all the vegetables and then give it to the cooks who would do the real work. And so it was a little dull. And I asked my boss, well, can I do something else? And he says, well, sure, as long as your customers are happy. And also make it something that's really, really important, because you already have a job, so just try for something really crazy. And so I told him, well, I'm going to design a protein then. And he says, well, you have to understand how proteins fold, and then you have to design them. That's a tall order at Anfinson. I remember him saying that. And what I didn't tell him is I really want to design an enzyme. Okay, so the way so here I am, I have some time, and I have peptides. I have a lot of peptides, and I thought, looking at the structures of proteins, that you could think about, say, forming this 4-helix bundle as just assembly of 4 helical peptides. And we could have a minimal assembly. We can describe the structure with parameters with D2 symmetry. And at this point, I met and started a collaboration with David Eisenberg. Here you can see him as a graduate student in 1968, and he's building a model of a protein, and he's using models from Linus Pauling. And these stick models were exactly the same stick models that we used to design our first protein. Now, I was really happy, though, after leaving his lab during a design session, going back to DuPont, taking the coordinates and putting it on a computer. This was much greater and more fun than dealing with these Tinker toys, and allowed us to use energy minimization and interactive graphics to begin to really design a sequence. So there I am with my computer. Now, the first peptide that we made to self-assemble is shown here, or one of the first, and it's composed of hydrophobic residues in green that are all the same, leucine, and polar residues that are charged to electrostatically stabilize the structure and give it water solubility. Now, what was the structure, though? At that time, because of the redundancy of the sequence, we were unable to solve it by NMR. But as we've heard now, we have AlphaFold. And in our lab, just the last month or so, we've also moved more to Chi-1, which is especially good for predicting complexes, and especially ones with metal ions in our hands. But it's very similar to AlphaFold 3. So now we can take that and say, what is the structure that we had designed? And what we find in high confidence is precisely exactly what we had designed. So we have the hydrophobic residues, they're packed just as we had thought, so this is great. Now, without knowing that, though, we had continued on to design a full protein by taking loops between helices, designing them to make a monomolecularly folded structure. And I remember being so excited about it. We made this protein and it was incredibly stable. And I went to a Gordon Research Conference, and it was my first of this type of a very scientific conference. And I got to meet a Nobel Prize winner for the first time. And I told him all about this, and he says, I don't know what you have done, Bill, but this is not a protein. I said, oh, okay. Why not? He says, because humans cannot design proteins. And so that left an impression, and it really motivated me tremendously. And also, when I came back, I started to say, in what way is this not a protein? How is it different? And this is the kind of a really detailed analysis that hadn't been brought to de novo protein design before. And what we found was that the interior side chains of the protein were more molten than those of native proteins. And now in the modern era, when we predict the structure, we see it's the same structure, but it's a very low confidence structure. So the predictions are sort of reflecting what we had seen. Well, having understood the problem, we know the solution. So we heard from Alan that as we fold a protein, it goes into a set of intermediates, and then ultimately one or so wins out, and we have a native state. So with alpha-4, what had happened was we went into this ensemble of intermediate-like structures, which was very, very low in energy, but not unique. To make it unique, what we had to do was to introduce some specific interactions and maybe decrease the stability a bit. And we were able to do this by taking some of those hydrophobic residues and turning them into ligands, histidines, that could then bind zinc. And we could show by NMR that the metal binding site was formed. We could show that it did, in fact, form a protein, but we didn't have the absolute fine structure at the time. And now when you put it into Chi-1 or AlphaFold 3, wonderfully, you see that it was precisely the structure that we had designed. So this was really the first de novo protein with a native-like fold. It was also the first type of a switch in a protein, because as you could see, we had zinc controlling both the topology and the dynamics. It was more dynamic until you bound the zinc. So the other way that you can control structure in proteins and uniqueness is to think about the residues in the core of the protein, and to make a selection of different sized residues so that they fit together in a unique conformation. And an important step towards doing that was from Michael Levitt and Joel Janine. And what they showed was that each of our amino acids have different shapes. They have different conformers, but there's only a certain number of them shown here. There's peaks there. And so that means now that we can treat each of these digitally as a different species. And so now you start thinking, already we're start thinking more like language models than a continuum. And that was incredibly important. Fred Richards could then say, hey, look, here's a protein of known structure. What residues could fit at each of these core positions in the protein? And what he found was a list, but he also found that that recapitulated what is seen in nature. So this is a very important contribution. Next, Desjardins and Handel developed a genetic algorithm, and now what you can do is try essentially all of the amino acids in all their low energy rotamers and see which collection really fits the core. And they made the protein and showed it worked. And then finally, Dayat and Mayo automated the process and showed that it worked for both the inside and outside of the protein for a 30 residue peptide that was previously a zinc finger but no longer required zinc. So we then used this method to design a 3-helix bundle similar to the 4-helix bundles in process. And now we have used Handel's algorithm to create a diverse set of amino acids. We had in all 19 different amino acids in the protein. And the NMR structure we solved at the time was really bang on. So this is incredibly validating. And so it was great. So we had the first de novo globular protein whose tertiary structure and sequence wasn't designed from scratch and who was validated. Now that became a really useful protein for many things. Because of its simple structure and its very fast folding near the speed limit, we were able, various people were able to study this to understand the landscape by which proteins fold. Others said, let's use this as a scaffold to build metal ions, and they were able to build metal ions that catalyze reactions with pretty good rates. Finally, a company that I'm not affiliated with called Arcellx said, let's use this beautiful de novo protein as a starting point, and then we can build side chains on the surface and use that to bind antigens, and that became quite useful for them in CAR T therapy, in which you're really programming a person's T cells to recognize cancers. So that was the first de novo protein to reach human trials, and it is by far the most advanced in terms of a therapeutic protein. There have been, on the other hand, vaccines made, but this is the most advanced in terms of therapeutics, and they have a nice pipeline. So this was very cool, and we were excited about it. Also, we were able to show that we could take this same motif, but make it 2 helices that recognize a 3rd helix. In this case, it's calcinurin's calmodulin binding domain. And we could show it was very specific because if we took myosin light chain kinase's calmodulin binding domain, it didn't bind. Now, all these years later, using Chi1, we can say, what did it look like? And it's superimposable. It's really quite remarkable how well it worked. So this was the first de novo design of receptors for natural peptides with high specificity and nanomolar binding affinity. Now we next, with David Eisenberg, went back and thought about designing some new folds that we hadn't seen in nature. And one of these was this domain-swapped 3-helix bundle. And I was a little worried when they came up with this design because it takes anions, or the cationic end of these helices and makes them bang against one another. And it binds anion, though, in the crystal structure that they solved. We also were able to use crystallographic symmetry to build larger assemblies. Here is a hexamer with two-fold and three-fold axes combined. And then finally, we were able to design entire crystals. You can think about this one here as having every atom in its structure specified by about an angstrom over millimeters. So it's amazing. And we can also position carbon nanotubes. In the area of membrane proteins, going all the way to 88, we had started designing proteins that served very selective proton transport or ion channel transport. Again, let's see what they look like today with AlphaFold 3 and Chi1, and they're superimposable. So we very much did nail the structures. Moving further into the future, we were able to design proteins that take protons and exchange them for zinc, and so we can drive zinc transport with a proton gradient or vice versa. And these were crystallographically and NMR characterized. Finally, we are interested in metalloproteins. As we heard, they're very important. And here's a large selection of di-iron proteins that had become available in the years after our design. And these are out of nature. And in their active site is a primitive 4-helix bundle, and you can bring that even further down to an alpha helix. And now we can use a parametric equation to basically describe the entire active site of this protein. And so that creates a mathematical embedding, which you now can use to design the entire protein from scratch. And so this has every atom is defined by a parametric equation. The side chains are put in using a repacking algorithm or sequence design algorithm, as we call it now. And when we solved the structure, we were just delighted to see that it was identical to the design. So that was the first high-resolution structure of a de novo metalloprotein as well. We were able to take that metalloprotein and change its chemical reactivity to either a 2-electron process or a 4-electron process, depending on how we changed the active site. And just to make sure that we could do things other than helical bundles, for example, here we have a mimic of rubridoxin, and Margaret Dayoff, back in '66, looking at the sequences, thought that there should be some symmetry in there, and indeed we were able to use that symmetry to create single-chain rubridoxin mimics, quite different from what it is seen in nature. So what did we do in these early years, the formative years? We defined the interactions required for folding. We introduced metal binding switches, specific peptide binding, and peroxidase catalysts. We had membrane proteins and ion channel function. We were able to make a 3-helix bundle that now is in phase 3 human clinical trials for cancer and looking very good. And we were able to use symmetries to build assemblies. So, but I think enough history. So what I'm really excited about is what we're doing now. And so there have been a number of advances. We've heard about several of these. One of them is diffusion, which was first published by Gavor Grigorian and Ingraham, and then about a week later by the Rosetta Group. And what one does there is one starts with a whole PDB full of structures, and then we noise them. We add Gaussian noise to the coordinates. And what that allows us to do is to have a model in which we know we can go backwards. So we know the gradients, the derivatives that we need to go from something that's collapsed to something that looks very much like a real protein. We can also only partially denoise the coordinates and use that as a starting point for design. So we have a lot of flexibility. Once we have a backbone like this, the next step, so that's starting noised, and here it's denoising. And Here we can now design a sequence, and this is done informatically at the level of just identifying the amino acids, and then the final step is always adding those side chains that correspond to the amino acids. And using that approach, Generate Biomedicine have already put molecules or improvements on antibodies into the clinic. So what we were interested in, because I'm a chemist that loves molecular recognition, is how do small proteins bind small molecules, and it would also be useful. So we started based on a suggestion from a cardiologist that he'd like us to design a protein that would neutralize the activity of Eliquis, a drug that's commonly used, because he'd like to get them into surgery quickly. And, but we can also use these to engineer drug-like properties for carriers and formulation. You could think about putting a receptor binding domain onto a small molecule binding protein that could then target to the right cell. And then if it gets endocytosed, it would start to get degraded or have low pH, and the drug can come out. So there's a lot we could do with this, but can we design proteins? Now, the way it's done right now by most labs is using neural nets, and these are very useful methods. But this is ligand MPNN, but generally it's a low probability event. You have to make tens of thousands of genes for a given small molecule you want to bind to, and then redesign it, and ultimately you can get something that is not as high affinity as the molecules that I'm going to tell you about in a minute. So we thought, well, how does nature do this? Nature basically has little I think of them as privileged positions where they can bind in the backbone, say, here's a phosphate, so that's a polar group, hard to dehydrate, but if you get it right, you can get some free energy of binding. And so then when you build a whole small molecule binding site, then you start to put these together in a modular way. So the real thing that you have to do is to find them and use them in a design algorithm. And so we used a device called the Vandermeer, which explains, given that I have a protein structure here, how it can bind a small molecule's polar groups in this case. We also now have Van der Graafs that tells us not just this backbone, but the overall environment. And so then when we're thinking about designing a drug, like this is apixaban or Eliquis, we can think about how can I really totally engage all the lone pairs and hydrogen bonded groups of this entire drug here. And we always focus on the polar things right, because if you don't get those right, you don't have a molecule that binds specifically or tightly. And then you can think about putting them in different scaffolds and find where you can satisfy all those functional groups. Having done that, we follow a workflow which is unique only in the last couple steps. And here, what we do is we predict what the structure of our protein is after we've made the sequence design. And then we say, is that pre-organized exquisitely well to bind the drug, which we can then see by docking calculations. And then by, most importantly, we use molecular dynamics simulations, which uses a physics-based force field rather than informatic approach. And you'll see how that plays out. So the first thing we did was a few years ago, we made 6 proteins instead of 10,000, and 2 of these worked. Retrospectively, we found 2 of them just didn't even fold, and we didn't require any optimization experimentally. And now we were able to show that it did exactly what we wanted to. If this is apixaban and we inject it into a mouse, now if we add the protein, it speeds up the clearance because it binds to it, and it's very rapidly cleared in the kidney and comes out in the urine. But you have to start to worry about immunogenicity with these artificial proteins So Bobo Deng at Westlake University made the all-D version of this. So it's a mirror image of it. And so that's going to have much, much less immunogenicity, in part because it can't be broken down into little peptides that are then presented via MHC molecules to create an immune response. And then this shows if we work really hard with our all-L form, we can get immune response, but with the all-D, nothing's doing. We just couldn't get one. So another collaborator was Alan Ashworth, who said, hey, do you think you could bind PARP inhibitors? I don't have time. I won't tell you about the biology, but I'll show you what they look like. And they have a common feature here. And what we wanted was a molecule that would bind all of these. And we needed to bind it much tighter than our original thing. And so what we did was satisfy, again, all the hydrogen bond potential, then add hydrophobic residues, build second shell interactions, and predict the structure in advance. Then we also looked at MD of molecular dynamics of both the drug and the drug-free form to make sure that it was pre-organized and not too dynamic. When we looked at it, we were very pleased to find the KD was less than 5 nanomolar for the molecule that we designed, and lost some affinity as we became less and less specific or similar to the drug rucaparib. And they worked in cell assays. Now, what I like most, though, is that starting from first principles, so this is a molecular dynamics simulation-based method that uses mean force to compute the ensembles we need to get free energy. We're able to predict in absolute terms the free energy of binding to within 1 kcal per mole. So that's about a factor of 2. And that's all done based on chemistry, real chemistry and physics. And again, we could show that this molecule worked even better for clearing the drug. So we've been able now to design many different small molecule binding proteins, many for more synthetic reasons. And we find that physics-based force fields are really very much in harmony and work together with bioinformatic methods, and with physics-based methods now having a big influx from machine learning. I think these are going to really be very complementary and sort of merge. And finally, the injection of some simple physical and chemical principles, if you learn them, into all these modern workflows, I think far outperforms unbiased and unguided uses of the algorithms. I would be really happy. My goal is to say that I have an algorithm where you can give me the drug and I'm going to find you a protein that's going to bind it with whatever affinity you want, just like you can order up an oligo that's going to bind to a gene. We aren't there yet, but I think we are there if we have some understanding of chemistry and physics. So now let me tell you what we're doing right now. And this is a project where previously I told you we looked at sequence diversity. So we design a protein, then we say, what sequence can we find? And that's a huge search of sequence space. But what if we now think about chemical space in parallel with sequence space? And what if we sort of walk through both of these landscapes at the same time? And that's what Yuta has done. And so we start with the apixaban binding protein ABL. We know that it's a very specific for binding to apixaban, but might this be a generalist for binding to other molecules? So to ask that, what we did was to work with Jamie Fraser, and he screened about 500 compounds, and he has them at 5 to 10 millimolar. And then he determines crystal structures and sees which ones bind and where do they bind. And what he sees is a hit rate from this that's very similar to natural proteins. And that was one of the questions we wanted to ask. Now, this shows all the fragments superposed. And one of the coolest things is this is the site that we thought might work. And this tyrosine makes nice aromatic interactions, but if that tyrosine just flips over a little, this isn't even a different rotamer, it's just slightly different, then we have a cryptic site that we never knew existed. And so we go back to MD, we can say, oh yeah, that did exist, it was just at a low population. But now can we use that? Could we bind something in that spot? And one of the molecules that bound there was a coumarin. And we said, well, could we do a fluorogenic turn-on coumarin? And that's, so to do that, we have to add a lot of bulk, extra groups. And so that means you go back and you redesign. So here's our apixaban binder. Where am I? Here's our apixaban binder. Here is the coumarin. And now if we add repack this, now we can be able to bind the turn-on. And so that's precisely what we did, and that's precisely what we learned. We were able to bind with high specificity for this. But let's return now to chemicals diversity space and say, let's challenge this molecule, this protein that we just designed, and see what else binds. And so we do that precisely, and this shows the ones that show some turn-on, and all of them look almost identical. So we kind of hit a fitness peak for binding this molecule. And now these have between 48 and 120 fold turn-on increase in fluorescence. And we're looking at more useful single molecule dyes, but that'll be another story. We also look at the affinity of these molecules and ask, is it as good as it can be? And to do this, we use something called ligand efficiency. You look at the free energy of binding and divide it by the number of heavy atoms, and that's because as molecules get bigger and bigger and bigger, they have more opportunities to make interactions, and therefore the affinity you can get, it goes up and up. And what we find are ones that are ligand efficiencies that are better than a lot of natural protein ligand pairs, and certainly better than our starting Abel protein. So, and then the crystal structure was solved and was exactly like the design. So now, can we design something that catalyzes a reaction? And just to get calibrated, the first thing we do is start with one that other people have studied extensively, and this is a Kemp eliminase. And so people have designed proteins that catalyze this very simple reaction here, and they call them enzymes. And so we went and said, let's take this transition state analog and put it right into the binding site. And now let's complete the design. We have to have this base, and then we have to hold on to it. And so now what we find is that the protein we designed is 100-fold better than any of the many, many proteins that had ever been designed using de novo algorithms. So that's pretty good. Now, they also use directed evolution, which you just kind of randomly change all the residues around the site or all over the site, and asked, how much, if we use that, how much can we move up the catalytic efficiency? And we get to a value of 10 to the 5th per molar per second. We said enough's enough because it's sort of an exercise. And others can reach that, but it takes about, in this case, 17 rounds. And what's different here is that they were always starting with active sites of real native proteins and sort of cheating that way, and then trying to build in the binding site, whereas we're starting entirely from scratch. Okay, so what did we learn, though, when we look at that triple mutant with an increased affinity, is a proline appears right in an alpha helix. And a tryptophan here appears and pushes the helices apart. But you think of this as a helix breaker, but what it does is helices are sort of quantized in the way they stick their amino acids out, and this allows you to sort of move that last half angstrom or so that you need to get catalysis. If we look at molecular dynamics simulations to find how is this base interacting with the transition state analog, what we can find is before directed evolution, we have a reasonably dynamic holding on of the substrate, but it becomes very tight after just this one round of directed evolution. We can also look at the angle, or let's look at first at the distance that it approaches, and you can see that it's very close to what is ideal from quantum mechanical calculations. And the angle is also ideal. And then most importantly, the reason that this reaction works so well with a carboxylate is that you dehydrate the active site, and you can see that we've done a very, very good job of dehydrating the active site. So again, what we're learning is injection of chemical common sense can do very well for these benchmarks. And directed evolution is one way to sort of get that, I call it the last angstrom problem, in that we can get very close in our designs, but getting that last angstrom where things really have to be perfect is something that we haven't done yet, but I think we will. And it's nice to have simulation to help us with that. Okay, so let's think about what we've done. We've started in amino acid sequence space in this dimension here and chemical space there. And we go up a fitness landscape. So here's cable. That was the catalytic one. Here's the one that binds apixaban, and there's the one that binds fluorescent molecules. And now let's think about chemical sequence space, and you can see these are in different spots. They're all helical bundles. They're all fairly similar, but together they really span a large distance in this landscape. And so then we find them at peaks in the landscape. But model reactions are useful because we learn a lot. So we study things like coumarin esters and nitrophenyl esters and this Kemp reaction in our lab and others. But really to go the last step, we have to do something useful. And something that would be useful in the lab and certainly in plants to be much greener catalysts. And that's what I'm going to finish telling you about. And so we sat down and said, okay, if we want to make an enzyme, what do we want out of it? Well, we'd like to use both artificial cofactors as well as natural cofactors that can be put in by biosynthetic machinery. We'd like it to be small so that it could be atom efficient. We'd like our substrate concentrations to be the type that we use in chemical plants and in our round bottom flasks. So that means millimolar to molar. And that's a big difference from what we're doing already in sort of model reactions. We'd like them to be evolvable in the event that we don't get it perfect the first time. They should be thermal stable. And finally, they should be stable, we think, to green solvents like ethanol, because a lot of our substrates just aren't very soluble in water that we would like to work with. So having some organic solubility would be good, but had never been demonstrated. So that brings us to sort of the thought, especially the concentration. We don't really need to bind tightly. When you're at 1 molar small molecule drug, that drug is sort of banging on the active site constantly by thermal motions. So you don't need to concentrate it. You do need to position it, though. And indeed, if you bind too tightly, then that's going to put you in a really deep energy well, as Jeremy Knowles told us, and then you have to climb out of that. So we want it to be weak binding. And we want stereoselectivity over many substituents. So the reaction we started with is one that Frances Arnold made famous in the biocatalysis area, in which she takes a styrene here with a double bond and a carbene precursor, and these two come together, and when they do, they create 2 chiral centers. So we have 4 diastereomers, and our job is to only make one of them in stereoselective catalysis, or stereospecific. And this is a collaboration with Yang at UCSB and Peng Liu at University of Pittsburgh. Yang being an excellent organic chemist and biocatalysis person, and Peng Liu, multi-scale modeler. And so we designed proteins that took an artificial porphyrin and we put in a transition state, starting with one of our older proteins, and then we say, how can we bind to it? And so we elongate this helix and build a loop. it didn't work that well, so we go back and we solve the structure and we think, oh, Lord, this is what happened. The helix really didn't keep going like it was supposed to. Our diffusion algorithm wasn't all that good. And when you go back and you sort of think about it, of course, if you're using an algorithm out of informatics, it's going to be paying attention to a lot of different things. And what you really want is to be sure that it's really, really specific. And so we went back actually to very old method, master, to get the ideal structure just for this loop. And when we did that, the first 4 proteins that we made out of 10 showed greater than a 99-to-1 enantiomeric ratio. So that's good. It's making 1 isomer. We could also show that at 0.25 molar substrate, so reasonable concentrations, we get 40,000 turnovers. So it's really behaving like a very good enzyme that you would use for biocatalysis. So this matches the most successfully engineered natural proteins that have been made. It's stable up to 70% ethanol, and that's the data to prove that there. And we also can do the same with a silane here. It's a silicon hydrogen insertion reaction, and we get 3,000 turnovers. And this time, though, we used a natural heme. The great advantage there is you don't have to insert it, and you can really screen for this reaction in whole cells. And so that's what we've done. And we also then did directed evolution and got to 10 times the best engineered natural enzymes ever. And it's stable up to 60% methanol. And when we looked at what happened with directed evolution, once again, we found a proline right in an alpha helix, really fine-tuning that active site. Again, we could look at molecular dynamics of the TAC complex and see that our protein was holding the substrate in just precisely the desired orientation to maximize the rate of the stereo-selective reaction. We've also done much more with membrane proteins, but I've run out of time. And so what I'd like to do is just conclude, again, a plea to keep rational intelligence as we use our AI tools, because as they become more powerful, we really need to stay one step ahead of them so we know what to ask them to do and make best use for society. And I think proteins will be increasingly practical solutions, particularly for the environmental applications I mentioned. And these are all the people that ever worked in my lab, 100 postdocs to date and 44 graduate students. They come from all over the world, although only a fraction of them worked on protein design in the lab. They all contributed to the environment of the lab, and I appreciate them all. Thank you. Okay.